Bismillahirrahmanirrahim. Today, inshallah, we shall talk about the scientific method. It's one of the most important topics, actually. We shall start by considering the following. You want to know why earthworms leave their burrows after it rains? See, if you go outside after it rains, you'll see earthworms everywhere for some curious reason. The question is why? Well, it turns out that in fact earthworms, they breathe through their skin. That is, as long as their skin is moist, they get the oxygen that they need directly from the air. It diffuses into the skin directly from the air. But when it rains, the water percolates into the ground and the water surrounds the earthworms. They're basically drowning in the rainwater underground. And there's not enough oxygen dissolved in the rainwater for the earthworms to get their oxygen that they need. So when it rains, the earthworms come out of the ground so they can actually breathe oxygen from the air. Well, that's what we believe now, but apparently there was a time that they used to say, well, the reason you see earthworms on the ground after it rains is that there are horse hairs on the street in the ground, and when it rains, the horse hairs, they turn into the earthworms. So that was the theory. Clearly, we don't believe that to be the case anymore. But this example illustrates two very important things. One, you should be observant about the environment around you. You should have a certain natural curiosity about the environment around you. As Sherlock Holmes said to Watson, Watson, you see, but you do not observe. Not only do you have to observe, you have to ask questions. See, why did the earthworms come out of the ground? And then you have to do something else, and that is you have to try to figure out why they do, and you have to design an experiment to test your theory or your hypotheses. Now this leads us to our topic of the day. Ibn Haytham rahimahullah is credited actually with developing the scientific method. See, in the old days, like in the times of Aristotle, for example, if Aristotle wanted to come to an answer about a particular question, he would use logic, he would use philosophy, he would think through the problem and come up with an answer. In other words, he would think through the earth-world problem and try to figure out an answer. Now, the scientific method, in fact, which is credited to Ibn Haytham, is one of the greatest contributions, one of the greatest gifts to the scientific community. See, the scientific method says that if you have a problem that you want to solve, you come up with a question, you come up with a hypothesis, and then you design an experiment to prove or disprove your hypothesis. That is the beauty of the scientific method. There is no logical proof, there is no philosophical proofs, there is simply a hypothesis for which you come up with a creative experimental design and you test your hypothesis. That is the beauty of the scientific method. The scientific method emphasizes on experimental data, and experimental data that can be reproduced either to strengthen or weaken the hypothesis. Now, here is an outline of the scientific method that he proposed. See, the scientific method starts with a certain observation. All knowledge really starts with observation. For example, why is it that the earthworms come out after rain? So when you have an observation, you have to formulate a question from it. Why is this so? And once you have a question, you have to come up with your best guess. And that's your hypothesis. The best guess may have been that it's a horsehair that turned into earthworms or something else, but that's a hypothesis. It's better than nothing. So when you observe something, you come up with the question, try to explain it, and that's your hypothesis. And then you have to design an experiment to test the hypothesis, to prove or disprove the hypothesis. The experiment gives the results, and the results can be interpreted and conclusions can be made. But the beauty and the importance of the scientific method is that it does not end with the conclusion of this one experiment. The strength of the scientific method is in its emphasis to repeat the experiment by the individual who performed the experiment and by other individuals to make sure that the methodology was correct. For example, somebody could do an experiment and come to a particular conclusion, but the laboratory equipment in there is very inaccurate, and somebody else tries to reproduce it and they get completely different results. So the scientific method places an emphasis on performing experiments and performing repeated experiments to strengthen the information, to strengthen the conclusions to which one arrives. So this is the scientific method. In short, the scientific method starts with observation, which leads to hypothesis, which results in an experiment from which you can derive results in conclusion, and then you repeat the process. Here's another example that illustrates a very important point. Apparently, the brown tree snakes were accidentally introduced into the island of Guam in the 1940s. See, here's Guam. There's the north of Australia over here. I don't quite know what they mean by accidentally introducing. Oops, like I lost a snake type of thing. But somehow the snake got introduced into this island. And initially, people were quite excited about the phenomenon because they thought, the snakes will eat the rats and the mice and the rodents of the island and make the place a more habitable place, perhaps. But it so happened that biologists went to the island, and this was apparently the second visit, and they noticed that, yes, the snakes were there, but somehow the birds were disappearing because they noticed that the sounds of the birds in the forest were quieter. So they said, man, this is quite an amazing phenomenon. This place is not nearly as loud a bird sound that it was. Now, to me, this is a very remarkable thing because you think this individual had to remember the forest sounds before and after, and this individual had to observe that there was a change, right? It's like there's a difference between seeing and observing. Now, then they had to propose a hypothesis, and they said the snakes were eating the birds, or more likely the snakes were eating the eggs of the birds, right? So they proposed an hypothesis. So this illustrates a point that you have to be aware of your surroundings. You have to see, you have to observe what's going on in your surroundings. You can't just walk through a particular area and never see what actually surrounds you, right? So the nature of science is that you have to always be curious, you have to always be observant, and you have to always ask questions and speculate on potential answers and hypotheses. But the hypotheses that one generates, they have to be testable. In other words, you can't come up with a silly hypothesis. For example, you say, one day the snakes decided that, they're going to eat the birds, or one day the birds decided to leave the island because they didn't like the island anymore, or something like that, right? So hypotheses that one comes up with, they have to be testable hypotheses, than the ones that could be transformed into experiments. Here's an example. The best of experiments, there are many different types of experiments, but the best experiments are controlled experiments. What we mean by controlled experiment is that you design an experiment where not too many things are happening, and you can actually test a particular hypothesis by narrowing down the variables. I'll explain this to you. For example, let's say you get a plant food, like over here. It says you use this plant food and with this fertilizer and whatnot, that your plant will grow exceptionally well. Well, the question is, how do you know that to be the case? Really, right? It's a claim, right? It's a claim. Maybe they're like kind of ripping you off or something like that. But how would you design an experiment to test that? To test the claim of the Miracle Grow, for example, that the plants will grow better if you use their product. For example, if you were to bring this Miracle Grow and put it on the plant, and the plant grows wonderfully, how do you know it was the Miracle Grow? But maybe because you're paying more attention to the plant and watering it more, right? So how would you design an experiment? Well, the way you would do that is first you would come up with a hypothesis, right? The hypothesis to test this would be, right, that the fertilizer helps the plant grow. The hypothesis here is the fertilizer helps the plant grow. Now, see, the best of hypotheses are stated not like a statement like you have over here, fertilizer helps plant grow, but they're stated as a conditional statement, as if and then. A conditional statement, if you recall, is a statement that has two parts. The first part is if blank, and the second part is then blank. So what you have to learn to do is formulate your hypotheses as a conditional statement. And that's not very difficult to do after a little bit of practice. That is, in this case, if a plant receives fertilizer, then it grows better. If a plant receives fertilizer, then it grows better. So you have to start thinking and formulating your hypothesis as conditional statements, because those are very, very useful. Then you can design an experiment, see? So what you do is, in a controlled experiment, so there, as I said, there are many different types of experiment, but in controlled experiments you have two groups. So here you have a plant, in this case plant B. You're not going to mess with this plant. You're not going to give this plant the fertilizer, see? Plant A here is going to be the one that's going to get the fertilizer. So you have one group here and a second group here, right? Or you can say one group here and one group here. This group here, which is not going to receive the fertilizer, that's called the control group. And this group over here, this plant here, that's going to receive the fertilizer is called the variable group. So the variable group is going to receive the fertilizer. The one that's not going to receive anything is called the control group. Now the reason the control group is important is because what I suggested. How do you know the fertilizer actually helped plant growth? Maybe the plant grew because you're watering it more, because you're paying more attention to it or something, right? But if you have a control group, you say, no. See, I had these two plants, okay? I watered them equally. I gave them enough, both the same amount of sunshine. They both got good quality soil, right? But this one got fertilizer and this one did not, and therefore this one did better. Therefore, we have to conclude that plant A did better because it had the fertilizer. So that's the value of the control group. Okay, the only difference between the control group and the variable group, the only difference between these two groups in the best of designed experiments, is that the variable group has one variable that's different. That's why it's called a variable group. That's one thing that's different or variable between the two. Otherwise, these two have to be identical for the experiment to be picture perfect. In other words, one cannot have a better soil than the other. I mean, you can't give this the best soil ever, and you can't give the cruddy soil muddy over here or something like that. No, they have to have the same soil, the same amount of water every day, the same amount of sunshine every day, etc., etc. So both of these two groups have to be identical in every way, except for the one thing that you're testing, and that is the variable. In this case, the fertilizer that this plant is lucky enough to receive, so to speak. Okay, and then you can chart their growth. Yeah, so the variable is the one thing that is being changed between the two groups, and then you plot the growth and you observe this. Then you can conclude, okay, well, here obviously you're taking measurements, right? Now look, this is a very interesting graph. Here you're charting weeks and you're checking the height, right? Why is the height important? The height is important because that's how you're measuring the growth, right? How are you going to measure plant growth? Are you measure it by the number of leaves? That's one way of measuring the height, right? That seems like a logical thing to do. So the variable is the fertilizer, and what you're actually measuring, the effect is the height. So the variable is the one thing that you are changing between the two groups. Now when we speak of variables, we talk about independent variable and dependent variable. Independent variable is in fact the condition that is being tested, that is the fertilizer. That's the one thing that's independent between the two groups. This will make more sense when I'm talking about this. The dependent variable is the result of that particular change, right? So the fertilizer, it's different between the two, and what does the fertilizer do? It causes the plant to grow. So the plant growth is presumably dependent on the variable, okay? And again, the only difference between the control group and the variable group is everything is the same, the soil, water, sunlight, etc., except the variable group has one thing that you're testing over here, the fertilizer. I want to give you another example of this. See, if somebody were to test, for example, a cholesterol medication, you would have a control group where they do not get the new cholesterol medication, and then you had a variable group that gets the cholesterol medication. In fact, you give the control group a placebo, so they're thinking, they don't even know, because you could have a placebo effect, people so you give them a medication as a placebo, like a sugar pill, whatever, so they think, they don't even know what they're getting. So the variable group, it gets the actual medication, the control group does not get the medication, and then you can compare the two groups to see if the cholesterol medication actually works or does not work. So it's very important to have control group. In that case, the variable would be the medication, right? The independent variable would be the medication, and the dependent variable would be the cholesterol level. Now, the beauty of the conditional statement is if you write your hypothesis as a conditional statement, it helps you identify the independent and dependent variable, because the independent variable always comes first, dependent variable always comes second. If a plant receives fertilizer, that's the independent variable, then it grows better, that's the dependent variable. So independent variable is the fertilizer, plant growth is the dependent variable. So if you write the hypothesis as a conditional statement, then it becomes very easy to identify independent and dependent variables. So as we review the scientific method, it includes the following. It all starts with observation, which leads to a question, which is just a hypothesis that you come up with. That's your best guess as to why things are the way they are, which helps you to design a creative experiment to test that hypothesis, from which you get results, and the results can give you conclusions that you can derive from those results, and you have to repeat the experiment to make sure it's reproducible, and that strengthens the merit of the conclusions that you make. And that's the strength of the scientific method. Now, allow me to suggest you two things here. First, let's see if you do an experiment. Okay, if you do an experiment, you have a certain hypothesis, and you do an experiment, and you come up with a certain conclusion that you believe to be correct. So, are you now ready to make a theory about the phenomena? Can you make a theory with the phenomena, about the phenomena? So let me repeat that again, because it is so important. Let's say if you do an experiment and you have a particular hypothesis about a certain phenomenon, and you come to certain conclusions, and then you're very happy about it. Are you now ready to formulate a theory about the phenomenon? The answer is no. You can't come up with a theory based on one experiment. See, a theory in science is a very heavy word, whether you call it evolutionary theory or theory of gravity, etc. You don't use the term theory unless you have done boatloads of experiments about the phenomena, unless you've done a lot of experiments. So when scientists say the theory of evolution, what they're trying to imply is they have done a lot of groundwork. It's not something that's very casual. So you have to do many experiments to reproduce the evidence to come up with a theory. So one experiment does not give you a theory. One experiment basically gives you one experiment. Okay, that's extremely important. And has suggested that all starts with observation, and hypotheses that you generate have to be testable hypotheses. They cannot be opinionated things, right? So, like, who's the coolest cricket player or something? Who's the coolest basketball player? And that's an opinion. You can't really test that as a hypothesis. You know what I'm saying? So, hypotheses have to be testable. When we do experiment, there in biology, there are two types of experiments particularly. Okay. There are two types of experiments particularly. Experiments from which you gather numerical data, these are called quantitative experiments. Okay, quantitative experiments are experiments from which you get number data. Okay, so for example, over here, these are paramecium unicellular organism. See, this experiment is about paramecium survival rates. So the way you read a graph again is you first read the title, paramecium survival rates, and then you read the x-axis, then you read the y-axis. So this is a graph about paramecium survival rates, which compares the temperature and the number of paramecium surviving. As the temperature increases, the paramecium, they grow pretty good. So they like kind of toasty temperature over here, but when it gets really hot, they start to die off, right? So there's a spectrum of temperature right over here that they kind of really like, okay? But here, this is a quantitative experiment. You're actually measuring numbers. You're measuring temperature. You're measuring number of paramecium surviving. So these type of experiments where you gather numerical data, these are called quantitative experiments, which give you quantitative data. Then there are experiments that are qualitative. Here you get descriptive data. This is what Darwin did. He took he described many phenomena, and so when you're describing things, because you can't really get quantitative data on things that have gone extinct. You know, how are you going to do that? and 65 million years ago is when dinosaurs went away. So descriptive data is called qualitative data, and those experiments are called qualitative experiments. So these are two different types of experiments. Biology is particularly important because in biology there are a lot more qualitative experiments than in other sciences such as chemistry and physics, which tend to be more quantitative in nature. In talking about getting data, or quantitative data, numerical data, we talk about two different concepts. One is accuracy and the second is precision. I'll explain this to you. By the time I'm doing this, I think inshallah you understand this. Accuracy is how close a measurement is to the accepted value. In other words, how correct it is. This, for example, this is a pretty accurate target here. Because if this being the correct value, they're pretty close to being correct. Now over here, this is precision. Precision is how close a series of measurements are to each other. I mean, how reproducible, how consistent the results are. Now this individual, they're kind of off the target, but they're at least consistent. You know what I'm saying? So this is high precision but low accuracy. Right? The best thing would be if you had high accuracy and high precision, right? So this person is accurate, right? This person is very precise. So accuracy is a measure of correctness. Precision is a measure of consistency of the data. I'll give you another example to drive home the point. So this individual is accurate. I mean, they're pretty close to the target. I mean, they're authentically close to where they need to be. Now this individual they're not accurate, they're nowhere close to where their target is, but they're precise. I mean, they're pretty consistent. They're messed up, they're like shooting over there, So they're not accurate, but they're very precise. Now this individual, it's like Michael Jordan here, is accurate and precise, right? So they're close to the target and they're very consistent in their measurements, so to speak, right? So this is the difference between accuracy and precision. So you want to be the best experiments, of course, are those that generate accurate and precise information. Most scientists, internationally at least, use the metric system, which is the decimal system in taking measurements, and we typically use the international system of measurements, or most commonly use the international system of measurements, again, which is a decimal system where length is measured in meters, mass in kilograms, time in seconds, and electric current in amperes, and temperature in kelvins, compared to, for example, Celsius. That's not a international system measurement unit. And amount of substances in moles, and intensity of light in candelas, and volume in liters. So this is typically the measuring system that is used internationally. And finally, we should talk about science and society, because these are introductory remarks on the science of biology. Can scientific knowledge be ethically neutral? Is science entirely always ethically neutral? Some people would say so. Some people would suggest that science is neutral. How we use is determining its moral inclinations. Well, one can say, well, is the science of warfare, is it really ethically neutral? I don't know. So we have to be careful when we say science is always neutral. I'm not certain that the science is always neutral about things. And second thing is, can science answer all questions? I think no, science cannot answer all questions in life. We can throw science at trying to answer all questions, but does not always work. And this is very important because people think that you can solve world poverty by doing more scientific research. Sometimes it's just you just have to open up your heart and just do the right thing. Science is not always the answer to all questions. Some human problems are sometimes more complex and sometimes quite simple. It just takes the willpower to solve them. Thirdly, can science solve all problems? Can we solve all human problems technologically? The answer again is no. But don't underestimate the value of science or what it can do for us. Just consider the water treatment plants. If it weren't for water treatment plants, you wouldn't have clean drinking water to drink, and we don't give a second thought about that. But some 2 billion people in the world don't have clean drinking water. And the vaccination that you probably took as a child protect you from measles, mumps, and rubella, and polio, etc. So they've saved millions of lives. And antibiotics, penicillin, which is considered penicillin, or the ear infection, or the bronchitis, or the strep throat that you might have had, that you were lucky enough, blessed enough to get antibiotics for. High yielding crops and labor saving devices. So science has done many, many good things. It's a blessing from Allah subhanahu wa ta'ala, and no doubt about it. But we do also have to be careful how we use it, because science also has had negative effects. For example, excessive use of fertilizer has caused the beautiful coral reefs to disintegrate, and the abundant, beautiful life underwater is actually disappearing. And the pollution that has resulted from consumption and waste is also a negative effect of science. And of course, the effects of global warming is of great interest. In this regard, I think the science of biology is more important now than ever before in trying to rescue our planet. However, you can get despondent, but human beings are greater than some of their challenges. So the problems that we create, we can also solve. Until next time, Assalamu alaikum.